Hey there, and welcome back to the deep dive. Today, we're really getting into the engine room of the cell. We're talking about how your body creates the energy that powers, well, everything.
That's right. We're tackling oxidative phosphorylation and mitochondrial function. It sounds like a mouthful, I know.
It definitely does.
But our mission here is pretty simple. We want to make it clear how the energy in your food gets turned into usable fuel, ATP. And crucially, what happens when that system runs into problems.
Because this isn't just abstract biochemistry, is it?
Not at all. It has huge implications for health, disease, even aging. Understanding this is really key.
Okay, so think of this as a trip inside your cells, right into those tiny power plants, the mitochondria. We're going to figure out the how and why of energy production. Let's start with the big one. What exactly is oxidative phosphorylation? The energy equation fueling the cell.
Okay, so at its heart, oxidative phosphorylation is your body's main strategy for making ATP, adenosine triphosphate.
The energy currency, we hear that a lot.
Yeah. Exactly, it's the universal fuel. This process takes the energy unlocked from breaking down food, glucose, fats, you name it, and packages it really efficiently into ATP bonds.
So the energy from my lunch isn't immediately ATP?
Not directly, no. Most of it, after the initial breakdown steps, is captured in what we call reduced coenzymes. Think of them as tiny, rechargeable batteries.
Okay.
The two big players are NADH and FAD2H.
Right, NADH and FAD2H. So they're like energy shuttles,
Yeah.
carrying electrons.
That's a perfect way to put it. They are electron carriers, loaded with potential energy. They ferry these high-energy electrons to a special system inside the mitochondria,
And that system is?
the electron transport chain, or the ETC. It's embedded in the inner mitochondrial membrane.
Okay.
These carriers, NADH and FAD2H, drop off their electrons to the ETC. The electrons then get passed down a line of components, like a tiny bucket brigade.
And where do they end up?
The very end acceptor is oxygen, O2. The oxygen picks up the electrons and gets reduced to water, H2O.
Ah, so that's why we need to breathe oxygen.
Fundamentally, yes. And here's the crucial bit. As the electrons move down that chain, energy is released. It's not wasted, though.
Right. It's used for something.
It's used to power the addition of a phosphate group onto ADP, adenosine diphosphate, turning it into ATP. And this amazing molecular machine called ATP synthase does that job.
ATP synthase. Got it. So what's the efficiency like? What's the payoff?
It's remarkably good. For every NADH that gives up its electrons, you generally get about 2.5 ATPs.
Wow.
And for FAD2H, it's around 1.5 ATPs. This makes oxidative phosphorylation by far the biggest ATP generator in your body. It's essential. The chemiosmotic model, the cell's hydroelectric plant.
Okay, that's a huge amount of energy, but I'm still trying to picture how just passing electrons along makes ATP. You mentioned the chemiosmotic model.
Yes, and it's really quite brilliant. Peter Mitchell won a Nobel Prize for figuring this out.
Okay, so what's the idea?
Imagine the ETC components, those protein complexes, sitting in the inner mitochondrial wall, the membrane. As electrons move through specific complexes 1, 3, and 4V, these complexes act like pumps. They use the energy from the electron flow to actively pump protons.
Protons, hydrogen ions. Right.
Exactly, H+. They pump them from the inside compartment of the mitochondrion, the matrix, out into the space between the inner and outer mitochondrial walls, the inner membrane space.
Okay, so you're building up a high concentration of protons outside the matrix, like water building up behind a dam.
That's the perfect analogy. You create this steep gradient. There are more protons outside than inside. And because protons are positively charged, the outside becomes positively charged relative to the inside.
So it's both a chemical gradient and an electrical gradient.
Precisely. Together, that's called the electrochemical gradient, or the proton motive force. It's a powerful stored energy source. The protons really want to flow back into the matrix, down their gradient.
And ATP synthase lets them.
Yes. ATP synthase provides the channel. You can picture it like a tiny molecular turbine embedded in that membrane. It has a channel, the F0 part, that lets protons rush back through.
F0.
And as they flow back into the matrix through F0, they cause a part of the enzyme to literally spin.
Spin, like a motor.
Exactly like a tiny rotor. This spinning shaft connects to the catalytic part of the enzyme, the F1 headpiece, which sits inside the matrix. The rotation causes the F1 part to change shape. And these shape changes are what drive the synthesis of ATP from ADP and phosphate, and then release the newly made ATP. It's called the binding change mechanism.
Wow. So it is like a tiny hydroelectric dam and turbine.
It really is, harnessing the flow of protons to generate rotational energy, which then does the chemical work of making ATP. It's beautiful. 3, the electron transport chain components, the molecular relay rays.
All right, that model makes sense. The gradient powers the turbine. Now let's look closer at the chain itself. Who are the actual players passing these electrons along? You mentioned complexes.
Right, it's a specific sequence. The electrons don't just jump randomly. They're passed between various components, each with a slightly different ability to attract electrons, what we call reduction potential.
Okay.
So NADH typically hands its electrons off to complex I, which is also called NADH.CoQ oxidoreductase. It's a huge complex. It contains things like flavin mononucleotide, FMN, and multiple iron-sulfur, FES centers. These help shuttle the electrons along within the complex before passing them to coenzyme Q.
Iron-sulfur centers, does that relate to things like iron deficiency?
Absolutely. That's a great clinical connection. If you're iron deficient, like ANR from one of the case studies, you might not make enough functional FES centers or enough of the cytochromes we'll get to later. This impairs electron transport, reduces ATP production, and contributes to that feeling of fatigue.
Makes sense. Okay, so complex I passes electrons to coenzyme Q. What about FAD2H?
FAD2H comes from sources like the TCA cycle, specifically from the enzyme succinate dehydrogenase, which is actually part of complex 2.
So complex 2 is part of both the TCA cycle and the ETC.
Yes, it's unique that way. Complex 2 also passes its electrons directly to coenzyme Q. An important difference, though. Complex 2 does not pump protons. Only complexes 1, 3, and 4 do.
Okay, good distinction. Now tell us more about this coenzyme Q. You said it's special?
It is. CoQ, or ubiquinone, is unique because it's not locked into a protein complex. It's a small, lipid-soluble molecule that can diffuse freely within the membrane.
So it's like a mobile carrier.
Exactly.
Right.
It shuttles electrons between complex 1 and complex 3, and between complex 2 and complex 3. It can also carry protons, which is key for proton pumping.
Interesting. But you also mentioned a downside, free radicals.
Yes, because it can exist in a half-reduced state, a semiquinone. It's prone to accidentally passing an electron directly to oxygen, creating superoxide, a damaging reactive oxygen species, or free radical. The cell has defenses, but it's a known hotspot for generating these.
Okay. So after CoQ, where do the electrons go?
They go to complex 3, the cytochrome BC1 complex, and then to cytochrome C, another mobile carrier, which ferries electrons to complex 4.
Cytochromes. You mentioned iron again.
Right. Cytochromes are proteins containing a heme group, similar to hemoglobin, with an iron atom at the center. But here's the key difference you asked about earlier.
Okay.
In hemoglobin, the iron stays in the F2+ state to bind oxygen. In cytochromes, like BC1CAA3, the iron atom cycles between the oxidized F3+ state and the reduced F2+ state. That's how it accepts and then donates an electron.
So it has to change its charge state to work in the ETC.
Precisely. It's a fundamental functional difference.
Okay, so complex 3, cytochrome C, and then complex 4. What happens there?
Complex 4 is the end of the line. It's also called cytochrome C oxidase. It contains cytochromes A and A3, and also crucial copper ions. This is where the electrons are finally handed off to molecular oxygen, O2.
And oxygen gets turned into water.
Yes, it takes 4 electrons to fully reduce 1 molecule of O2 to 2 molecules of H2O. Complex 4 carefully manages this 4-electron transfer.
And all along this path, complex I, 3, and 5e protons are being pumped out.
Correct. Electron transport is tightly coupled to proton pumping. You can't have one without the other running normally. Complex 3, for instance, uses a clever mechanism called the Q cycle to help move protons across the membrane as it transfers electrons.
So what happens if the protons aren't used? If ATP synthase isn't running because the cell has enough ATP?
The proton gradient builds up really high. This creates what we call proton back pressure.
Right.
That back pressure makes it harder for the complexes to pump more protons out, which in turn slows down the rate of electron flow through the chain and consequently slows down oxygen consumption. It's a feedback mechanism.
Okay, that makes sense. But what if something blocks the chain itself? Like cyanide?
Exactly. Cyanide is deadly because it binds very tightly to the iron in complex 4, specifically cytochrome A3.
So it blocks the very last step.
It blocks the transfer of electrons to oxygen. This causes the entire electron transport chain to back up, like a traffic jam. No electrons can flow.
Which means no proton pumping.
No proton pumping anywhere along the chain. The proton motive force dissipates almost instantly. ATP synthase stops working because there's no gradient to drive it.
And the cell runs out of energy very quickly.
Extremely quickly, leading to cell death. This is why cyanide poisoning is so rapid and devastating, and it connects back to that clinical example of Cora N. Her nitroprusside treatment, while helpful initially, carry that risk of cyanide toxicity if used too long, highlighting how critical uninterrupted electron transport is. 4. Energy yields and cytoplasmic NADH shuttles.
That really drives home how vital this chain is. Okay, we talked about the ATP yields, 2.5 for NADH, 1.5 for FAD2H. Is there anything else about the energy involved?
Well, the overall process releases a significant amount of free energy. That's what makes the electron flow energetically favorable, essentially downhill. It's irreversible under normal conditions.
And does all that energy go into ATP?
Not quite. It's estimated that maybe only about 30% or so of the energy released from oxidizing NADH or FAD2H is actually captured in ATP bonds.
Only 30%. Where does the rest go?
It's released as heat. The ETC is actually a major source of your body heat. This energy release helps maintain your body temperature.
Ah, that makes sense. So inefficiency isn't always bad.
In this case, it's physiologically useful. And this again ties back to Cora N's heart attack. Severe ischemia means very low oxygen.
Right, the final electron acceptor is missing.
Exactly. So the ETC stops, proton pumping stops, ATP generation plummets. That lack of ATP is a major reason why the heart muscle cells get injured and can die if oxygen isn't restored quickly.
Okay. Now, you mentioned earlier that NADH made outside the mitochondria, in the cytoplasm, can't just wander in. How does its energy get utilized by the ETC?
That's a really important point. The inner mitochondrial membrane is impermeable to NADH, so cells use shuttle systems.
Shuttles? Like little ferries?
Sort of. They don't move the NADH itself, but they transfer its electrons across the membrane. There are two main ones.
Yeah.
The glycerol 3-phosphate shuttle is prominent in tissues like muscle and brain. It takes electrons from cytoplasmic NADH and ultimately transfers them to FAD within the inner membrane, which then passes them to CoQ.
So it feeds into the chain via FAD.
Right, which means cytoplasmic NADH using this shuttle only yields about 1.5 ATP, similar to mitochondrial FAD2H.
I see. What's the other shuttle?
The malate aspartate shuttle. It's more complex, found in liver, kidney, and heart. It effectively transfers electrons from cytoplasmic NADH to generate mitochondrial NADH.
Ah, so it results in NADH inside the mitochondria.
Exactly, which means cytoplasmic NADH using this shuttle yields the full 2.5 ATP. It's more energy efficient.
So the total ATP we get from, say, a molecule of glucose depends partly on which shuttle system the cell is using for the NADH made during glycolysis.
Precisely. If glucose is completely oxidized aerobically with oxygen to CO2 and water, the total yield is typically cited as 30 or 32 ATP molecules per glucose. The difference depends largely on which shuttle handles the cytoplasmic NADH.
And compare that to anaerobic glycolysis.
Which only yields a net of 2 ATP per glucose when it ends in lactate. It's a huge difference.
Wow. That really highlights why oxygen is so critical for getting the most energy out of our food.
Absolutely. To make the same amount of ATP anaerobically, a cell would have to burn through glucose roughly 15 times faster, which is what happens in tissues during severe oxygen deprivation, leading to that buildup of lactic acid. V-regulation and the concept of uncoupling.
Okay, this is a remarkably efficient energy factory, but how is it controlled? How does the cell match ATP production to its actual energy needs? Can't just run full tilt all the time, right?
No, definitely not. It's tightly regulated. And the beautiful part is the electrochemical gradient itself is a key regulator. It couples the rate of the ETC to the rate of ATP synthesis.
How does that work?
It comes down to the availability of ADP and inorganic phosphate, pi. When you use ATP for cellular work, muscle contraction, thinking, whatever, you generate ADP and pi.
Right, the breakdown products.
High levels of ADP essentially signal to ADP synthase, hey, we need more ATP. ADP binds to the synthase, which then allows protons to flow back into the matrix faster, making more ATP.
And the increased proton flow lowers the gradient.
Exactly. The proton motive force decreases slightly. The EDC senses this drop and says, uh-oh, gradient's falling, need to pump more protons. So the EDC speeds up, consumes more oxygen, and pumps protons faster to rebuild the gradient.
Which then stimulates the breakdown of more fuel.
To supply more NADH and FAD2H to the now faster-running ETC.
Yeah.
It's a beautifully coordinated feedback loop.
So when I exercise, I use ATP, oxygen use goes up, ADP synthase speeds up, proton flow increases, the gradient drops slightly, the ETC speeds up, oxygen use goes up, fuel burning goes up. and I generate more heat.
You got it. That's exactly the chain of events. And when you rest,
The opposite. ATP levels are high, ADP is low, ATP synthase slows down, fewer protons flow, the gradient builds up high,
creating that proton back pressure.
which slows down the ETC, slows oxygen consumption, and slows fuel use.
Perfect. That's called respiratory control or acceptor control,
Huh.
since ADP availability is key.
Okay. Now, you mentioned heat generation. What about this idea of uncoupling?
Right. Uncoupling happens when the link between electron transport and ATP synthesis is broken. Specifically, it means protons leak back into the mitochondrial matrix without going through ATP synthase.
They bypass the turbine.
Exactly.
Hmm.
The ETC might still be running, pumping protons out, consuming oxygen, burning fuel. But if those protons just leak back in elsewhere, the energy stored in the gradient isn't captured as ATP.
So where does that energy go?
It's dissipated primarily as heat.
So uncoupling generates heat without making useful ATP energy.
Precisely. This can happen chemically. Certain lipid-soluble compounds, like dinitrophenol, a notorious diet drug from the past, or even high doses of aspirin, can shuttle protons across the membrane, acting as chemical uncouplers.
But does it happen naturally?
Yes. Physiologically, we have uncoupling proteins, UCPs. These are specific proteins that form channels in the inner mitochondrial membrane, allowing a controlled leak of protons back into the matrix.
Why would the body want to do that?
The classic example is a UCP1, also called thermogenin. It's abundant in brown adipose tissue or brown fat.
Brown fat. I've heard of that.
It's especially important in newborns and hibernating animals for non-shivering thermogenesis. When they get cold, signals trigger the release of fatty acids in brown fat, which activate UCP1. Protons leak back through UCP1, generating a lot of heat to warm the body up without needing muscle shivering.
Fascinating. Are there other UCPs?
Yes, UCP2 and UCP3 are found in many other tissues. Their exact roles are still being researched, but they might be involved in regulating metabolism, maybe protecting against oxidative stress by reducing the proton gradient slightly, not just generating heat.
And this connects to the clinical case of Stanley T with hyperthyroidism.
Exactly. Stanley T had Graves' disease, leading to high levels of thyroid hormones. Thyroid hormones are known to increase the expression of UCPs, particularly UCP2 and UCP3.
So his mitochondria were more leaky.
Effectively, yes. His basal metabolic rate was high, his ETC was running fast, consuming lots of oxygen, but because of the increased uncoupling, less of that energy went to ATP and more was released as heat.
Which explains his heat intolerance and sweating.
Perfectly. And it's also worth noting that even in healthy individuals, there's always a small basal level of natural proton leak across the mitochondrial membrane, unrelated to specific UCPs. This baseline uncoupling actually accounts for a significant chunk, maybe over 20% of our resting metabolic rate, because keeping the system slightly leaky costs energy. 60. Mitochondrial transport and cell fate, the gates of life and death.
Okay, so we've made ATP inside the mitochondrial matrix, but most of the cell's work happens outside the matrix. How does the ATP get out? You said the inner membrane is tight.
It is very selective. It requires specific protein transporters, or translocases. The main one for ATP is the ATP-ADP translocase, sometimes called ANT.
ANT, okay. What does it do?
It performs a strict one-for-one exchange. It moves one molecule of ATP from the matrix out into the cytosol, but only in exchange for bringing one molecule of ADP from the cytosol back into the matrix.
So it ensures the building blocks, ADP, get back in while the product ATP gets out.
Exactly, and interestingly, this exchange is actually helped along by the electrochemical gradient itself, because ATP has one more negative charge than ADP, so moving ATP out is electrically favorable.
Clever. Are there other transporters?
Oh, yes. There are transporters for phosphate, to bring pi in with the proton, pyruvate, and others. The mitochondria needs to import fuels and extra products. There's even a uniporter for calcium ions.
What about the outer mitochondrial membrane?
The outer membrane is quite different. It's much more permeable to small molecules and ions. This is because it contains proteins called porins that form large channels called VDATCs, voltage-dependent anion channels. Think of it more like a sieve.
Okay, so inner membrane, tight control. Outer membrane, more open.
Generally, yes. But things can change dramatically under stress. There's something called the mitochondrial permeability transition pore, or MPTP.
MPTP. That sounds ominous.
It can be. It's thought to be a large, non-specific channel that can form across both the inner and outer membranes, often at points where they touch. It involves components like the ANT we just mentioned, VDA from the outer membrane, and other proteins.
What makes it open?
Severe cellular stress signals. Things like excessively high calcium levels inside the mitochondria, high phosphate levels, lots of reactive oxygen species, or conditions like ischemia, lack of oxygen. Conversely, normal ATP levels and slightly acidic pH tend to keep it closed.
And if it does open, what happens?
It's catastrophic for the mitochondrion. The pore is huge and nonspecific. Protons flood back into the matrix, instantly collapsing the electrochemical gradient.
So no more ATP synthesis.
Worse, ATP synthase actually starts running in reverse. It uses up existing ATP, trying desperately to pump protons back out.
Oh, wow.
Ions and small molecules rush into the matrix, water follows, and the mitochondrion swells up like a balloon, often rupturing the outer membrane. This leads to irreversible damage and usually triggers necrotic cell death, the messy kind.
Is this related to that ischemia reperfusion injury you mentioned?
Very much so.
Hmm.
When you restore oxygen after a period of ischemia reperfusion, it allows the ETC to start up again, which is good for making ATP.
Blood.
But the conditions during reperfusion, like potential calcium overload that occurred during ischemia, pH shifts, and a sudden burst of reactive oxygen species from the restarting ETC, can actually trigger the opening of the MPTP in vulnerable mitochondria.
So restoring oxygen can paradoxically cause more damage in some cells.
Exactly. It's a major challenge in treating conditions like heart attack and stroke. Preventing MPTP opening is a big therapeutic goal.
And does mitochondrial damage link to other forms of cell death?
Yes, mitochondria are also key players in initiating apoptosis, which is programmed or clean cell death.
How so?
The inner membrane space, that area between the inner and outer membrane, stores several proteins that, if released into the main cell compartment, cytosol, trigger the apoptotic cascade.
Like what?
Things like cytochrome C, yes, the same electron carrier, procasposes, and other factors. If the outer mitochondrial membrane becomes permeable, perhaps due to damage, MPTP flickerings, or specific signaling pathways, the release of these factors signals time to die for the cell. 7. When the system fails, OXPHOS diseases and lactic acidosis.
It's clear the system is powerful but also vulnerable. What happens when the basic machinery itself is faulty due to genetics?
That leads us to OXPHOS diseases. These are a group of disorders caused by defects in oxidative phosphorylation, stemming from mutations in the genes that code for the necessary proteins. These mutations can be in either the cell's main nuclear DNA, nDNA, or in the mitochondrial DNA, mtDNA itself.
Wait, mitochondria have their own DNA?
They do. It's a small circular piece of DNA located right inside the mitochondria. It contains genes for just 13 essential protein subunits of the ETC and ATP synthase, plus the RNA machinery needed to make them.
13 proteins? That doesn't sound like much.
It's not, but they're critical. The vast majority, over 1,000 mitochondrial proteins, are actually encoded by your nuclear DNA, made in the cytosol, and then imported into the mitochondria.
Okay, so mtDNA is different. How else?
It has some really unique genetic features. First, it's inherited almost exclusively from your mother through the egg cell. Sperm mitochondria usually don't make it into the embryo.
So maternal inheritance?
Right. Second, during cell division, the mitochondria and their mtDNA are distributed randomly to daughter cells. If a mother has a mix of normal and mutant mtDNA, a state called heteroplasmy, her children can inherit varying proportions of the mutations.
So siblings could be affected differently.
Exactly. Symptoms often only appear when the percentage of mutant mtDNA in a particular tissue crosses a critical threshold. And because tissues like the brain, heart, skeletal muscle, and kidney rely so heavily on ATP,
They're usually the most affected.
precisely, they have a lower threshold for dysfunction. Also, mtDNA has a much higher mutation rate than nuclear DNA and poor repair systems, so mutations can accumulate, especially with age, partly due to damage from those free radicals generated by the ETC itself.
Can you give an example of an mtDNA disease?
Sure. A classic one is MERRF, which stands for myoclonic epilepsy with ragged red fibers. Patients might have muscle twitches, myoclonus, seizures, dementia, hearing loss, heart problems, a whole constellation of symptoms in high-energy tissues. And looking at the family history often reveals that clear maternal inheritance pattern. Muscle biopsies might show ragged red fibers, which are muscle cells stuffed with abnormal mitochondria.
Wow. What about mutations in the nuclear DNA that affect mitochondria?
Those also cause OXPHOS diseases. Since most mitochondrial proteins are nuclear encoded, there are many more potential genes involved. These typically follow standard Mendelian inheritance patterns, often autosomal recessive.
But they still mainly affect the high-energy tissues.
Yes, because even though the mutation is present in all cells, the functional deficit, the reduced ATP production, becomes most apparent where the energy demand is highest.
Is this related to the case of Isabel S, the patient with myopathy?
It could be. Her case illustrated how external factors can also cause mitochondrial problems that mimic genetic diseases. Certain medications, like the older HIV drug zidovudine, AZT, were found to inhibit the enzyme that replicates mtDNA.
So the drug depleted the mtDNA.
Yes, leading to acquired mitochondrial dysfunction, particularly in muscle, causing weakness and those characteristic ragged red fibers. It shows how sensitive mitochondria can be.
Okay, one last connection we kept hearing. Lactic acidosis. Why does that often pop up with these mitochondrial problems?
It's a common consequence of impaired oxidative phosphorylation. When the ETC isn't working properly, it can't efficiently reoxidize NADH back to NAD plus man.
So NADH levels build up.
Right. You get a high NADH-NAD plus ratio in the cell.
Right.
This high ratio inhibits key enzymes, like pyruvate dehydrogenase, which normally channels pyruvate from glucose breakdown into the TCA cycle.
So pyruvate can't be used efficiently by the mitochondria.
Correct. Instead, the cell converts the excess pyruvate into lactate using the enzyme lactate dehydrogenase. This reaction actually consumes NADH, helping to regenerate some NAD+, so glycolysis can at least continue.
Ah, so it's a backup way to keep some energy production going via glycolysis, even if it's inefficient.
Exactly. But it leads to lactate accumulation. Plus, the cell senses the low ATP levels and tries to compensate by running glycolysis even faster, producing even more pyruvate, which gets shunted to lactate.
So defective ETC leads to high NADH, which shunts pyruvate to lactate, and the cell ramps up glycolysis, making even more lactate.
That's the essence of it. So lactic acidosis is often a key indicator that cellular energy metabolism, particularly mitochondrial function, is seriously impaired, whether due to genetic OXPHOS disease, toxins like cyanide, severe oxygen deprivation, or even things like impaired pyruvate metabolism. Outro.
Wow. That was quite a journey into the mitochondria, and it's just incredible to think about all those moving parts, the electron carriers, proton pumps, that amazing spinning ATP synthase turbine, all working together constantly.
It really is a marvel of biochemical engineering, and hopefully this discussion highlights just how central it is, but also how delicate the balance can be. We've seen how genetic flaws, toxins, lack of oxygen, even subtle things like proton leaks can have really significant consequences.
From fatigue all the way to severe disease and cell death. So thinking about the listener, what's the big picture takeaway here?
I think it's appreciating this powerhouse within us. Understanding oxidative phosphorylation isn't just about memorizing pathways. It helps explain how we get energy, why oxygen is vital, how certain diseases arise, maybe even aspects of aging related to accumulating mitochondrial damage. It connects a lot of dots.
It definitely does. This deep dive really showed how these microscopic processes have such macroscopic effects on our health and lives. Thank you for walking us through that.
My pleasure. It's a fascinating and incredibly important system. We hope this has given everyone listening a clearer picture of that amazing energy factory inside their cells.
Absolutely. Well, stay curious, keep learning, and we'll catch you on the next deep dive.